So let's start. So it's a pleasure for me to introduce Werner Greene. So he's one of, ours. He's coming from UCSF, from the other side of the bay. He's an MD, PhD. He's a professor at UCSF. He's also the director of the Gladstone Institute for Virology and Immunology. And he's heading a lab, a really active lab, working on this molecular pathogenesis of, HIV malignancies. Essentially focusing on this accessory, what we call accessory proteins, so like VIF, NEF, and others. He received many accolades and recognition among his career. He's also the president of the Association of American Physicians. He's a member of the Institute of Medicine of the National Academies and a fellow of the American Academy for the Advancement of Science. So thank you very much. And
thank you. Thank you. Thank you. Nope, I'm not going to use that, so I don't need that. Well, it's a real pleasure to be here. I traveled significantly less far than any other speaker, I think, just across the bay. So in the next 35 to 40 minutes, I would like to tell you a story about how CD4 T cells are lost during HIV infection. But before that, let me try and frame the global HIV epidemic as we know it now. So 35 million infected, 36 million dead. Many would argue it's the third or fourth worst epidemic in human history, and this epidemic continues to expand, albeit more slowly than in the past. You can see that sub-Saharan Africa is bearing the brunt of new infections. Indeed, 70% of all new infections are in Africa. Now, I have been involved in the Accordia Global Health Foundation. Our work first started in Uganda, building an infectious diseases institute to try and curb the impact of HIV infection in that country. That country contains 36.3 million residents, 7.3% of whom are now HIV positive. It was in 2004 I made my first trip to Africa. I'd been working on HIV for probably almost 20 years. I did not understand HIV/AIDS until I went to Africa. One of the first things that I was confronted with getting off the plane in Entebbe was this. The Marabou stork, this prehistoric bird flying, and it's also named the Undertaker bird. And if you'd like a closer view, this is it. These are some of the ugliest things you have ever seen. And they will go into a dumpster. Oh my gosh. You don't want to be anywhere near that dumpster as they tear the thing apart. But also on the way from the airport, I also saw this. And that is one of the burgeoning industries in Uganda in 2004 was the making of coffins. Both large coffins and small coffins for all of the children and adults that were dying of HIV/AIDS. This is a clinic at the Infectious Diseases Institute. This was the occasion of the inauguration. I arrived Monday morning and went to the clinic on Monday afternoon, and I walked in. There were 300 people in the waiting room, and I could have dropped a pin and heard it. It was almost complete silence. And I thought to myself, whoa, that's kind of strange. I came back to the clinic the next day on Tuesday, and the clinic was abuzz. People were talking animatedly, the kids were running around. It was a completely different situation, completely, day and night difference. And I asked my host, I said, what's the difference between Tuesday's clinic and Monday's clinic? Oh, he said, it's very clear. Tuesday, all these patients here are ones that were lucky enough to enroll in the first antiretroviral drug trial at the Infectious Diseases Institute. These people have a hope of living. The people that came on Monday are slowly but surely dying. So I wrote a monograph, turning Mondays into Tuesdays, that kind of captured all of my impressions, which were amazing from this trip. And I've been involved. I subsequently became president of the Accordia Global Health Foundation, now executive chair. We continue to work steadfastly in Uganda, expanding to operations into Malawi and Nigeria, as well as creating an association of centers of excellence within within Africa. Of the 35 million people currently infected, based on new WHO criteria, 26 million of those people urgently need to be on antiretroviral therapy. We should be treating 26 million. But of that 26 million, we're only treating slightly around 10 million of the individuals, 9.6 million. Eric Gooseby would say this number is now closer to 11 million, but it's quite clear that the gap is immense, and there's no way that we really can double or triple our investment. So this is a disturbing statistic that while we're doing well, while we're making great gains, still there is so much to be done. Also, for every 10 people that are started on antiretroviral therapy in Africa, 16 people are newly infected. You can do the math as well as I can and see that we do not yet have a successful solution for the HIV crisis ongoing in sub-Saharan Africa. So it's always been in the back of my mind, is it possible to devise a safe and inexpensive therapy to stop the progression of HIV disease in the millions, now almost 17 million people in the developing world who currently have no access to antiretrovirals? Could we shelter in place? Could we provide a temporizing treatment that would keep them from dying of AIDS until they can get on antiretroviral medicines? So just park that idea in the back of your mind, and now let me tell you the story about how CD4 T cells die. So despite nearly 25 years of study, we still do not fully understand how HIV depletes CD4 T cells. This, of course, is the fundamental problem in AIDS. If you don't lose your CD4 T cells, you don't develop the immunodeficiency. Now, there are two principal death pathways that have been proposed. First, productive infection of CD4 T cells. For the longest time, and many in the field continue, thought that the major mechanism by which HIV depletes the cells is it infects those cells and kills them. But direct infection occurs within such a small, or this productive infection occurs within such a small fraction of the cells, it cannot come close to explaining the massive CD4 T cell losses that are being recorded. This led to the idea that maybe infected cells are somehow negatively influencing bystander cells in the neighborhood, leading to ill effects upon these uninfected bystander cells, and that these bystander cells might be really driving CD4 T cell depletion. It was within this kind of background that we started our studies and what we elected to do was to study how the T cells are dying not in blood, but in lymphoid tissues, where in fact 98% of CD4 T cells reside. The types of tissues that we used were the human tonsil and human spleen. Tonsil is a much more prevalent tissue. Spleen only becomes available when someone has a splenectomy, for example. The establishment of national tissue repositories was key for us being able to access these tissues on a regular basis. These studies have been led from the very beginning by Gilad Deutsch, now a staff research investigator within the Gladstone, really brilliant young scientist who has really accomplished a lot in these studies. Nicole Galloway, a biomedical sciences graduate student, has also participated critically within these studies, and I'll mention others as I go along who've contributed to the story. So here's the first experiment I'd like to present to you. This is a tonsil culture which has been infected with HIV. And we're monitoring here CD4 T cells. We're infecting with a virus that if the virus productively infects that cell, the cells turn green and they score in the GFP, or green fluorescence protein channel. They also, HIV infection also leads to downregulation of CD4. So these productively infected cells primarily occur in the upper left-hand quadrant, reflecting green but low expression of CD4. We see that by day 6, which is about when the peak of productive infection occurs, that only about 1.5% of these cells are GFP positive. The CD4 population here, these are the quote bystander cells, they appear to be quite happy. However, within 3 days later, there's almost a complete loss of these CD4 T cells. Now, certainly initially we had to exclude the fact that these CD4 T cells were becoming productively infected, moving into this gate and dying before we could score them. And that was pretty easy to do because we were able to label these CD4 T cells, these bystander cells, and then follow their fate in the presence of the productively infected cells. And I will now summarize a whole body of work that represents several years. What did we find? I'll summarize just by this schematic. So we have two types of cells in the tonsil. We have activated CD4 T cells, which comprise roughly 5% of the population. These are the cells that classically fuse the virus, reverse transcribe, converting into double-strand DNA. The DNA is integrated into the genome. New virions are made, and ultimately, after these virus-producing factories produce new virions, ultimately these cells die, generally by a caspase-3 dependent process of apoptosis, a very silent or bland form of programmed cell death, shown here, that involves the formation of these apoptotic bodies, which are engulfed by macrophages, which really limits the inflammation. So this is a silent or very quiet form of programmed cell death. In contrast, let's look at the other 95% of the CD4 T cells resident within the tonsil. Here we see, in fact, that these cells are also susceptible to the virus fusing, but because of their resting state, reverse transcription is impaired, and short pieces of DNA, incomplete reverse transcripts, or short pieces of DNA accumulate, which we went on to show that this DNA is sensed by the cell, and that the cell launches against that DNA an innate antiviral response, which leads to the assembly of inflammasomes, the activation of caspase-1, and ultimately a death by a process called pyroptosis. Now you may be asking yourself, what's pyroptosis? I asked myself that question too. So pyroptosis is an intensely inflammatory form of programmed cell death. It's mediated by the activation of caspase-1 in the context of an inflammasome. Caspase-1 processes pro-inflammatory cytokines like IL-1 beta and IL-18 into their bioactive forms, but in addition to the production of these inflammatory cytokines, ultimately pores form in the plasma membrane of these cells, and the entire cytoplasmic contents leak into the extracellular space, creating an intense inflammatory environment. So pyroptosis, pyro for fire, a fiery form of cell death in contrast to the silent, quiet form of death with apoptosis. So when we looked at we became very interested in what type of programmed cell death was occurring in these bystander cells that were becoming abortively infected. And we were quickly able to establish that drugs like efavirenz and AMD 3100, an entry inhibitor and a reverse transcriptase inhibitor, could rescue these cells from death, as could a caspase-1 inhibitor, but not a caspase-3 inhibitor, nor a caspase-6 inhibitor. Also, in terms of the release of cellular contents, we monitored that by the release of cytoplasmic lactic dehydrogenase. We can see that, in fact, the antiviral drugs prevent that LDH release, as does the caspase-1 inhibitors, but not the caspase-3 inhibitors. So indeed, we were blocking, the caspase-1 inhibitors were able to block the cytoplasmic pore formation and release of cytoplasmic contents. But we really, since these, no drug, no inhibitor is as specific as you would like it to be, we really sought genetic support for the involvement of caspase-1 and the inflammasome by an shRNA knockdown approach. But of course, we're using resting CD4 T cells here, which are incredibly hard to transfect, highly resistant to lentiviral infection due to the expression of SAMHD1 restriction factor. But unperturbed, Shengeng and the laboratory undertook development of a technique by which we could introduce lentiviral vectors encoding specific shRNAs into these cells. We borrowed from the literature that had discovered that HIV, or the SIV VPX gene product, is able to degrade that restriction factor, SAMHD1, that prevents the lentiviruses from thriving within the resting CD4 T cell. And we were able to open a gateway with VPX so that we could introduce our lentiviral vectors and get gene knockdowns and then monitor the effects of those knockdowns. Here's an example of the VPX, from SIV VPX in a viral-like particle achieving a knockdown. It's a transient knockdown of SAMHD1. Interestingly, at about 120 hours, SAMHD1 levels are return to normal as the VPX that was packaged within the virion is degraded, is metabolized away. So, but the gateway is open. We see that in the case of HIV, that in fact using these are the levels of knockdown here. Using caspase-1 a knockdown, we see that we can rescue CD4 T cells from death. We also can rescue cells from death with ASC, which is a inflammasome cofactor. But interestingly, caspase-3 knockdown has no effect, and knockdown of the NALP3 or NLRP3 subunit of the NALP3 inflammasome had no effect, suggesting that it was not this NALP3 inflammasome that was being assembled. So any evidence for pyroptosis actually occurring in vivo? So we really sought this. We were able to access lymph nodes from subjects infected with HIV at San Francisco General Hospital who were on no therapy. They had decreased CD4 counts and high viral loads, and we were able to access them because of the great help from Peter Hunt and Hiroya Hatano, who run a lymph node biopsy service at SFGH. And we were able to directly stain these cells right out of the surgeon would remove the samples. They would be immediately processed and stained. So here, so this is a lymph node. This is the germinal center. This is the marginal zone. This is the pericortical region. And we see that, in fact, that the proliferating cells, as marked by Ki67, are in the germinal centers. This is where the T cells and the B cells get together to make an immune response. We also see that that's where HIV P24. I just lost my light. Oh, came back. No. It's dead. Is there another pointer?
It's that part, yeah. There you go.
It kind of fades, see?
Well, this one is not as bright. You have to hit the top button.
Yes, okay. Work on that one. Okay, so we see that the HIV P24 is highest in the germinal center, but then if we look for where the caspase-1 is being activated, it's being activated in the T cell zone, the pericortical region surrounding the germinal center. It's also in this pericortical region that we can see IL-1 beta being produced, and it's in this pericortical region that annexin 5, indicative of pyroptotic cell death, or many forms of cell death, is occurring. So in fact, in geographically, spatially differentiated areas within the lymph node, we see this whole pyroptotic process playing out, whereby the virally produced cells in the germinal center are likely killing the surrounding resting CD4 T cells in the pericortical region. So, oh, you have another battery? Oh, thank you. Thank you. Yes. Great, thank you very much. So bystander CD4 T cell death occurring during HIV infection is mainly a suicide, not a murder. So it's an innate immune response against the virus that is occurring in these resting CD4 T cells that is accounting for most of the CD4 T cell depletion. It's only in that 5% of activated and fully permissive CD4 T cells where the virus is the actual, the murderer. In contrast, it's a host response gone awry in the resting CD4 population. And detection of pyroptosis also now provides a new mechanism that links the two signature processes in HIV infection. And that, of course, is chronic inflammation, and CD4 T cell depletion. So this idea of the host response gone awry, that we have this abortive infection occurring in lymph nodes, in lymphatic tissue. This leads to pyroptosis, the release of pro-inflammatory cytokines and cellular contents, which causes an marked inflammation. The inflammatory response elaborates chemokines that attract new, healthy CD4 T cells to the site, to the zone of inflammation, and the pathway is just repeated time and time and time again, leading to the progressive depletion of CD4 T cells in large numbers. So is it possible to break this vicious pathogenic cycle in vivo? Well, there of course is a vulnerability, and that is caspase-1. Caspase-1 is obligately required for pyroptosis. Caspase-1 inhibitors, we were glad to find out, have actually already been produced and have been tried in the clinic. One of those is VX-765, a highly selective, orally bioavailable caspase-1 inhibitor from Vertex. They tried it first in chronic seizure disorder. It's been shown to be, in that trial of chronic epilepsy, it was shown to be safe and well tolerated over a 6-week period of time in humans. They decided not to advance it for that particular application because while they saw effects, there was just not enough caspase-1 dependent inflammation occurring in the brain as a driver of the seizure disorder. Now, in our system, VX-765 works superbly well, just like the other caspase-1 inhibitors in both tonsil and in spleen, to inhibit CD4 T cell death. So does treatment with a caspase, now another key question is though, well, what about the possibility that if we treat with a caspase-1 inhibitor, could we be converting the abortive HIV infection that's occurring in those cells, blocking the pyroptosis, but what will happen to those cells? Might they become productively infected, in which case that wouldn't be really good news for using a caspase-1 inhibitor in the clinic. Well, we see here infection in the presence of the caspase-1 inhibitor that there is no increase in the number of productively infected cells occurring compared to no drugs alone. So in fact, there's not a massive shift of cells that are being rescued from pyroptosis to now becoming productively infected. And in fact, this is consistent with the fact that the SAMHD1 block is still in effect in these cells and that they're non-permissive to productive HIV infection. So we're very interested in the idea of using inhibitors of caspase-1, inhibitors of the inflammasome to interrupt this vicious cycle of HIV pathogenesis. But there were still a lot of unknown questions here. And one was, what's the DNA sensor that is detecting the incomplete HIV RT products and triggering this pyroptotic response? And this is really where the Berkeley connection begins. So Kate Monroe trained here at Berkeley, and fortunately she came to my laboratory. In addition, Zhiwan Yang trained here at Berkeley and came to my laboratory, so I'm really liking this connection. They worked on the sensor project together. We took an approach of making biotinylated DNA, and then using that DNA to dip into CD4 T cell lysates to see what might bind, and then purifying that DNA protein conjugates, and then looking ultimately by silver staining, electrophoresis silver staining, but more importantly, mass spec. The mass spec was done in collaboration with Nevin Krogan and Jeff Johnson at UCSF. And the top 6 hits that emerged out of the many runs on the mass spec were these proteins right here. The first 3 are ones that are involved in DNA repair. IFI-16 was a known inflammasome component, as was IFI-X. We also looked in the cytoplasm of resting CD4 T cells for what might be binding to that DNA. We saw that IFI-6 and DNPK1 which was a component of the Ku complex. So these two guys were prominent for us, as was a weak signal of IFIX. In contrast, proteins like AIM2, which was our lead candidate from the beginning, Sting, DAI, they did not appear to be binding to the HIV DNA. AIM2 is actually only very weakly expressed and the more recently identified sensor CGAS is not expressed at all within resting CD4 T cells. So we got really interested in IFI-16. Gary Nabel had just reported the possibility that DNPK1 was a sensor in activated CD4 T cells, but not resting CD4 T cells. So IFI-16 was first recognized as an anti-proliferative and DNA damage protein. It was then shown to be a double-stranded DNA sensor capable of inducing interferon via the Sting pathway. And then more recently, it's been shown to be the first single-stranded DNA sensor within mammalian cells. And we show here that, in fact, IFI-16 does, in fact, in our hands, bind to single-stranded biotinylated DNA. It binds much better to double-stranded, but it certainly continues to bind to single-stranded. So then we used the VPX technology to introduce shRNAs against IFI-16, and Kate did these studies that were just recently published. And sure enough, she was able to rescue with actually she's used 3 different shRNAs against IFI-16 and was able to rescue these cells from cell death. Conversely, when we knock down AIM2, when we knock down Sting, or when we knock down IFIX, or when we knock down DNPK1, there was no rescue. So in using 2 different approaches for shRNA knockdown, IFI-16 scored positively in all of the assays. So IFI-16 appears to be a DNA sensor that senses or detects the HIV, the abortive infection DNA, and then launches the assembly of inflammasomes, caspase-1 activation, and the pyroptotic pathway. It's interestingly that IFI-16 can form its own inflammasome. And the inflammasome involves requires ASC for its function. So we think that the inflammasome is actually formed by IFI-16 in conjunction with ASC that then recruits the caspase-1 and then processes the IL-1 beta and leads to the pyroptosis. So why has the pyroptotic cell death pathway been missed for 25 years? Why am I standing here 25 years after the discovery of HIV telling you about the major way that CD4 T cells die? You would have thought somebody else would have discovered that. Well, blood CD4 T cells were used in virtually all prior experiments. And then, so we asked the question, do these cells die by pyroptosis like lymphoid cells following HIV infection? And by we, I mean Isa Munoz Arias, who's right here. Also, another graduate student from Berkeley. She's a graduate student who's just now writing her thesis. But what did Isa find? What Isa finds is these are the blood CD4, what she will find is that blood CD4 T cells are highly resistant to HIV-associated pyroptosis unless they are co-cultured with lymphoid cells. So here's a tonsil, where virus-producing tonsil cells are mixed with target cells that are of tonsil origin. You get the predicted depletion of CD4 T cells by pyroptosis. If you do the same experiment with blood cells now, instead of lymphoid tissue cells, there's no depletion. If you say, well, maybe you have to activate the target cells, still no depletion. Yet these PBMCs are making sufficient virus, because if you mix them with tonsil, the tonsil cells die. But if you mix, yes, the tonsil cells die if the virus producers are the PBMCs. But now if you make the PBMCs the target cells and use tonsil as the virus producers, now the PBMCs die. So there's something about getting these 2 cells in the same culture dish that allows the PBMCs to die by this pyroptotic pathway. And what is the nature of their natural resistance to pyroptosis? How is blood different than tonsil or lymph node cells? So we first asked, do HIV virions fuse to resting blood cells at lower levels than tonsil CD4 T cells, resulting in decreased abortive infection? Maybe there's not just enough DNA there to be sensed. Well, in fact, when we use our BPR-BLAM fusion assay with tonsil and blood CD4 T cells, we see, in fact, similar levels of fusion between blood and tonsil. So the virus is getting in just fine into these cells. You can see that it's nicely, the signal is specific and it's blocked by AMD 3100. So then the next question is, well, is the cytosolic HIV DNA generated by the abortive infection sensed equally in blood and tonsil CD4 T cells? Well, imagine we were very interested in this result, in that shown here is blotting for IFI-16 from 2 donors of blood cells, and we see that in the resting state, we can't detect IFI-16. However, if those cells are activated with PHA and IL-2, we can detect the major isoforms of IFI-16. Contrast the absence of IFI-16 expression under basal conditions in the blood versus the high-level expression of IFI-16, the DNA sensor in tonsil. So does and then the question is, well, when we put the lymphoid cells together with the blood cells, is that inducing IFI-16 expression? Is that why they're now dying by pyroptosis? Well, and what Isa is able to show that the effect is we believe the answer is yes. We can see a twofold induction of IFI-16. There's about a twofold induction also of serine 536 phosphorylation of the REL-A subunit of NF-kappa B. So these cells are undergoing a low level of activation that is sufficient to allow IFI-16 to be expressed, apparently in sufficient doses to now allow the pyrotic pathway to proceed. So these findings underscore AIDS as a disease of lymphoid tissue, not blood. Blood isn't dying. They explain why the pyroptosis pathway of cell death in HIV infection remained undiscovered for 25 years. The wrong cells were being studied. Everyone was using it in blood, and blood cells don't die by this mechanism. So let me just conclude with a few comments about what kind of infection is required for the elicitation of pyroptosis. Do cell-free virions work, or is perhaps cell-to-cell viral transmission required? And this is studies that have been performed led by Nicole Galloway, a BMS graduate student, not from Berkeley. And what we'd previously shown is that when you separate the cells, the interaction of CD4 T cells, when they're allowed to interact together, when the virus producers and the target cells are allowed to interact together, there is depletion. But if the virus producers and the effectors are separated by a transwell, there's no depletion. So these data suggested that some type of cell-to-cell interaction was likely involved. We asked, is formation of a virological synapse required for killing? The synapse, of course, is composed of virological synapse composed of ICAM1 and its cognate partner LFA1. And Kate and Nicole were able to show that ICAM1 antibodies were able to, if you broke the virological synapse with anti-ICAM1 antibodies, you could rescue cells from cell death. Similarly, you could rescue cells from cell death with anti-LFA1 antibodies. So it appeared that the virological synapse was critical, like supporting a cell-to-cell interaction or a cell-to-cell transmission mechanism. Another approach to asking this cell-to-cell question is to increase the surface area of the culture. As you increase the surface area of the culture, it makes it less likely that the cells can participate in a cell-to-cell interaction. What would happen? Well, what we found is that when you started the cultures in a 96-well V bottom, you get very nice killing. It continues in a 96-well U bottom, but as you go to a flat bottom 96-well, 24-well, 12-well, in fact, if we culture under conditions where the cells are really quite dispersed and interact poorly with each other, the 12-well flat bottom culture, we see almost no CD4 T cell depletion in the cultures. So we've concluded that cell-to-cell spread, and I'm not, there's other data to support this, but it's now clear to us that cell-to-cell spread of HIV is required for the induction of pyroptosis. What is my time? I have what, 7 minutes? Oh, perfect. Then I'm going to go back one slide. So there have been 40 species of African monkeys infected with their own HIV-like virus. And in virtually every case, over the hundreds of thousands, millions of years of evolution, these viruses have come to terms with the lentivirus. And all of them, and the solution is not to suppress the virus, but to rather to alter the host response to the virus. So all of these non-pathogenic lentiviral infections, for example, in sooty mangabes and green monkeys, they're associated with high viremia. And if you incubate if you incubate activated CD4 T cells with these viruses, they can infect those cells and deplete them. But these animals do not develop AIDS, and they do not develop inflammation and immune activation. And our preliminary data show that they display little or no caspase-1 activation. So we think that the evolutionary, that's played out many times, the evolutionary solution to the problem posed by lentiviruses may well be to somehow cancel out the pyroptosis pathway that leads to this intense inflammation. So to some extent then, using a caspase-1 inhibitor would be chemically mimicking that type of evolutionary solution. So that then brings me to my summary. Abortive infection and innate immunity to viral DNA drives most of the CD4 T cell depletion. Caspase-1 dependent pyroptosis is a major active pathway producing inflammation. Vicious cycle of abortive infection, pyroptosis, inflammation, and recruitment of new cells is established. Blood cells are highly resistant to pyroptosis because they lack the sensor. Cell-to-cell transmission appears required. And evolution to non-pathogenicity may involve lack of caspase-1 activation. So I want to leave you with this concept, and that is cell death in HIV infection is all about the activation status of the cell. So that different activation states can lead to the utilization of different pathways of cell death. In a fully activated lymphoid or blood CD4 lymphocyte, these are the cells that undergo productive infection, no SAM-HD1 block, and these cells are the ones that die silently by apoptosis. Now, it's likely that this is occurring all the time in these non-pathogenically infected monkeys, but because it's a silent death, those cells are replaced by the thymus and CD4 T cell counts are maintained, no disease. Contrast that with resting lymphoid CD4 T lymphocytes in tissue. Here, in fact, these are 95% of the tissue of tonsil, spleen, and lymph node. Maybe we're guessing probably 50-50 situation in gut-associated lymphoid tissue, but it's these cells, because they undergo abortive infection, and they die by pyroptosis. And this is the highly inflammatory form of cell death. Contrast them with a blood CD4 T cell, which is the most resting of these cells. They undergo abortive infection just like the lymphoid cells, but they survive because they're not activated enough to express the sensor, and thus the pyroptotic pathway is uncoupled. So how might we conceive using caspase-1 inhibitors? Well, there's lots of caveats and lots of reasons that this would not work. But I still am intrigued by the concept of a caspase-1 inhibitor. If it was cheap, if it was safe, if it was effective, once a day dosing, ideally no cold chain, that it might be able to form a therapeutic bridge for those 16 to 17 million people who are not able to access antiretroviral therapy. It could be used as a monotherapy because it's targeting a host enzyme as opposed to a viral enzyme. It's more likely that these caspase-1 inhibitors will be used in conjunction with, or will be evaluated for their abilities to act adjunctively with antiretroviral therapy. Maybe even in the presence of full suppression of the virus, there is some residual inflammation that's occurring in HIV-infected subjects, maybe driving the early appearance of diseases of aging. Well, we're curious about whether or not caspase-1 inhibitors might modify that level of inflammation. And finally, we're very intrigued by the concept that the inflammation is playing a role in the maintenance of the HIV latent reservoir through IL-1 beta produced during pyroptosis, driving IL-15 production by macrophages, which is causing homeostatic proliferation of memory CD4 T cells, which harbor the latent virus. If that scenario is in fact true, and a big question mark there, we don't know yet, caspase-1 inhibitor, but if it is true, caspase-1 inhibitors might become part of one component of a cure cocktail. So these are three just potential, and I put question marks here with all of them. We just don't know how it's going to play out at this point. But these are testable hypotheses, and the time to testing of these drugs can be shortened greatly because caspase-1 inhibitors have already been in the clinic. I'd love to be doing a phase 2 trial now. So I wanted to acknowledge I've shown pictures of all the individuals with the exception of Steffi Sawinski, who is actually working in Kampala, Uganda now. Our UCSF colleagues, our funding sources, UCSF CFAR, NIH, the Giannini Foundation, the Sable Trust, the California HIV Research Program, and the CARE Collaboratory, of which I'm a member. And I want to thank you very much for your attention.
Really nice, thank you. I have a question, an interest about that. It's why do you think it's associated with cell-associated infection and not free infection? Do you think it's because cell-associated, it's directing multiple virion and you need a dose of DNA to be sent? Or is it something else that is associated with fusion between the two that is also participating?
So why is cell-to-cell transmission required for the elicitation of pyroptosis? I think it's a dose response. I think that cell-to-cell transmission is roughly 100-fold more effective than cell-free virions. And I suspect that you have to deliver enough HIV DNA into that cell to overcome the effects of TREX, which is a 5' a nucleotide, an exonuclease, which can degrade that DNA. So you have to get above the ability of TREX to degrade that DNA. So I suspect that's why cell-to-cell is required. We're testing that hypothesis now.
I have a comment and a question. As you know, probably, the pyroptosis was first observed with various bacterial pathogens. In fact, Kate Monroe, who you mentioned, who worked in Russell Vance's lab working on Legionella, this is an example of a pathogen that induces a robust pyroptosis, but as we know, it's really not a human pathogen, it's an accident. And it seems that most bacterial pathogens, maybe perhaps analogously to the other monkey SIV-like pathogens, have evolved mechanisms to prevent the induction of pyroptosis. And so it's consistent with HIV in the human population being, if not an accident, recent. And that the virus hasn't yet, well, either the virus or both the virus and or the host haven't moved away from that yet. Because there's a lot of ways that the virus can prevent that eventually, or the host will. But that's just my comment. But in the cases where you don't have pyroptosis being induced, the DNA may be inducing type 1 interferon through other pathways, such as the STING pathway. And there's recent papers suggesting that type 1 interferons suppress CD8 responses. So possibly the same mechanism that's leading in the tonsillar cells to pyroptosis is leading to type 1 interferon in the blood cells, which is then suppressing CD8 T cells.
Well, we know, in fact, that there is, even in the tonsil and spleen tissues, there is an interferon response that's occurring in addition to the pyroptosis. The interferon response is not apparently required for the pyroptotic response, in that we can block the interferon receptors and pyroptosis proceeds fine. Now, why would you would think that why does this thing go screwy? I mean, it should be a defensive response, right? It should be that this response should be trying to defend. I mean, the cell is committing suicide, a pyroptotic suicide, in an attempt to protect the host, presumably. It's foreign DNA. Why is it dying by this incredibly inflammatory pathway? It turns out that CD4 T cells are uniquely primed. They're primed in that, in general, for example, to get IL-1 beta processing by caspase-1, you need two signals, an NF-kappa B signal to induce pro-IL-1 beta synthesis, and then the signal for the assembly of the inflammasome to process the pro-caspase-1. CD4 T cells in these lymphoid tissues have already received signal 1. CD8 T cells have not. They still require two signals. So there's something so the CD4 T cell population is uniquely primed. And I think that's why it spins out of control. All they have to see is the virus, an abortive infection, nothing else required, and they just they go down this pyroptotic pathway. So if you give us 1 million years, we'll probably evolve to avoid this. But right now, I think it's at the heart of HIV pathogenesis.
Very interesting. Are splenectomized and tonsillectomized patients long-term non-progressors, and why don't we just take a bunch of tonsils and spleens out to block cell-to-cell pyroptosis?
Well, you'd have to take out all of the lymph. I mean, yeah. I mean, I guess I don't understand your question, Bill.
The spleen is a pretty big organ,
yes.
And you might expect that they would be relatively protective to be splenectomized and to have your tonsils taken out. And is there any evidence that that's the case?
No. I mean, no, because there's so much lymphoid tissue throughout the rest of the body. No. In fact, I'm sure splenectomy puts you at increased risk. What? Pneumococcal pneumonia, right? Yeah. I remember that from medical school.
Yeah, I have a question which is, I think, more social or epidemiological than biological or biochemical. When I look at the global prevalence of HIV, I realize that there is a gradation in prevalence as you move from Southern Africa upward. You know, we have more HIV in Southern Africa than in Northern Africa, and there is that somewhat a gradation as you move upward to Europe. Is there really an explanation for this?
Well,
are South Africans more promiscuous than Ghanaians, for example? Because we have, I think the prevalence in South Africa is about 30%. 20-30%. Then in Ghana, it's about 3%. Thank you.
You make a very good point about the incidence rates. Now, there has been some suggestion, and I've not personally studied this, that the clade C virus that is common in South Africa may be more readily transmitted than some of the other clades, which could be driving a higher incidence rate in South Africa. I don't know if that's the case or not, but that's what I've heard as an explanation. Probably the original cases, the tracing back where HIV came from chimpanzees, probably transmissions that occurred early in the southeastern corner of Cameroon, then spread throughout Africa as the truck routes set up. But you're absolutely correct that South Africa has been disproportionately hit with this virus, and maybe it's because of clade C.
I have a question on the latent reservoirs Do you think the pyroptosis mechanism could be used to activate and then flush these latent reservoirs so that the antiretrovirals would become more curative?
Well, I think so I will say the following. The field in general is looking for inducing agents that will activate the virus without producing generalized cellular activation in order to avoid a cytokine storm. I myself, having studied the transcription of HIV for many years, I really think that we're going to have some level of cellular activation in order to get the virus activated to a sufficiently high level. It's built. It's got NF-kappa B, it's built to respond only under conditions where the cell would be activated. It responds to the same signals. Now, that said, I don't think we want to induce pyroptosis. I think that's way too strong. We don't want cells dumping their cytoplasmic contents, et cetera. Maybe I should give that more thought, but my initial reaction would be not to go that direction. Too strong. Yes?
You mentioned the Vertex compound. I was wondering if you could comment on its clinical development and is there any evidence of stabilizing CD4 levels? Obvious questions, but
well,
where is it?
Vertex, caspase-1 inhibitors have never been tested in HIV-infected subjects. And the Vertex drug, as I understand it, and I've had meetings with Vertex, and I'm trying to interest them in a clinical trial. Actually, 2 clinical trials, the drug alone without antiretroviral therapy and the drug as an adjunct to antiretroviral therapy. As far as I know, it's sitting on the shelf with no application yet. There's not an obvious disease for which it's been shown to have great effect. I'd like to change that.
Oh, so there's no active clinical development in this setting?
As I understand it. But I hope that our work galvanizes a change in that area. Yes.
How do you correlate the? It's late. How do you correlate the extreme inflammatory pyroptosis with the lenti, the slow progression of HIV? That cycle, the vicious cycle, wouldn't it?
So that cycle plays out in the absence of treatment, and some people progress to full-blown AIDS within 2 to 3 years after HIV infection. Most people here in the United States and Europe be more like 10 years. But this is, and it's a true testament to the resilience of our immune system that the thymus can keep replacing, et cetera. But it's well known that the lymph nodes fibrose due to inflammation. Well, I'm suggesting that pyroptosis is probably driving that fibrotic response, et cetera.
Thank you so much, Dr. Greene, for speaking with us today. We're going to be taking a coffee break. For those of you who are presenting posters this afternoon, the easels should be set up, so if you can please set up your poster. And we are going to reconvene here in 20 minutes at 3:20. Thank you.